All right, class, welcome back. We are going to be going over the 7.3.1 study on the basics of Mendelian genetics. So, in the 1850s, an Austrian monk named Gregor Mendel conducted a series of experiments that changed the way we understand genetics and the inheritance of traits. His ideas weren't accepted until years after he died, but he is now known as the father of genetics. Before Mendel, people knew that traits were passed from parents to offspring, but they didn't know how. Two ideas were popular: the theory of blended traits and the theory of acquired characteristics. Mendel used ordinary pea plants to show that these ideas were false. And here we have a picture that demonstrates this blended theory, which was false. A tall and a short plant might end up with a medium plant. So we saw something similar to this in a previous video when we talked about dominance, incomplete dominance, and codominance. So this is what we're talking about is that pure dominance. So maybe if there was some sort of incomplete dominance, you might see something like this, maybe. But this was before we knew anything about any of that. And luckily enough, Mendel he discovered a lot about this, how these traits are transferred from generation to generation. All right, so two theories were popular at the time. All right, there's the theory of blended traits that stated offspring are a mixture of their parents. All right, so a tall and a short plant might have an offspring that is medium. And the theory of acquired characteristics, where traits that were attained or acquired during the organism's lifetime were passed on to the offspring. So, for example of that, the theory of acquired characteristics would be that a giraffe stretched its neck in order to eat, and this would make the giraffe's neck grow longer, and then when the giraffe had an offspring, it would pass on that long neck to its offspring. So Mendel was successful because he used the scientific method. He made observations and he asked questions. He began his work with this question: how are traits passed down from parents to offspring? So that'll be our first block down here. How are traits passed down from generation to generation? I don't know if I want to do that in blue. Maybe I'll change the color of that. Let's go with black. There we go. Match the rest of them. So why was he successful where others failed? Well, that's because Mendel used the scientific method. All right. He designed an experiment. He analyzed the results. He was very meticulous. He wasn't even a biologist. He was a monk. He was a trained physicist. So this wasn't even part of his formal education. He just decided to do this as a experiment on the side. So Mendel's experimental design. So we'll go down over here to test subject. So in order to study how parents passed on traits to offspring, Mendel had to choose an organism. He chose the pea plant. But why did he do that? So peas reproduce quickly, they're easy to grow, inexpensive. In addition, Mendel noticed that the pea plants had many traits that showed up in only one of two ways. For example, his peas produced only smooth seeds or wrinkled seeds, nothing in between. By the end of his experiments, Mendel had used about 28,000 different pea plants. So what organism did Mendel choose to study? He studied pea plants. Right? Why did he choose this? They were cheap, right? Easy to grow, right? They had easily observable traits. So these easily observable traits, like the smooth versus wrinkled seeds or the flower color, right? So we call those phenotypes. We talked about those before. Phenotypes are easy to see. So now we'll go on to page number 5. So crossing pure lines. When two pure lines are crossed, offspring all look the same. An experiment that produces reliable results must have a control group. Mendel started his experiments with a group of plants that all had the same version of a particular trait. Each group was what he called a pure line. By mating individual plants in each of his pure lines, Mendel made sure that the plants in the pure lines passed only one form of each trait to their offspring. The pure lines were Mendel's control group. He could compare plants in the pure lines with the offspring that resulted from crossing two pure lines. How did he do this? He allowed the plants to self-pollinate. When a plant self-pollinates, the sperm from one plant fertilizes the egg of that same plant. Now you may be thinking, plants have eggs and sperm? What? Well, we can watch this animation to learn a little bit about the sexual reproduction in plants.
Many plants reproduce sexually.
A pistil?
Flowering plants have female parts called pistils that contain the plant's female gametes. The male parts are called stamen and produce pollen, which contains sperm. For fertilization, the pollen needs to come in contact with the pistil. This allows the sperm to swim down the pistil and fertilize the egg.
Okay, so define pure line. So a pure line is a variety of a species that all have the same trait and whose offspring will only have that trait. So what does that mean? I'll kind of go over here, draw on the margins. So we talked about how we have dominant traits. So let's say T is equal to tall. And we have, so dominant alleles, and then we have recessive alleles. Little t would be short. All right. So if a plant was homozygous dominant, it's big T, little big T, which would give you a tall plant. If it's homozygous recessive, you get a short plant. Well, what happens when you breed tall plants like this with other tall plants like this, right? The only option, the only thing that you can get out of that is a tall plant, right? This tall plant that is homozygous dominant can only give, let's get my highlighter out, there we go, can only give big T's. It can only give, and when it's bred with another one that only has big T's, it can only make tall plants. And if you have two short plants with the little T's, their offspring can only be short. So the functions of these pure lines were to act as a control group. Oh, there's my blue, pops up again. All right. So the genotypes of the pure lines would be big T. So we would call that homozygous dominant, and little t, homozygous. So that's homozygous dominant and homozygous Recessive. All right. So these are the pure lines. Page 6. The first generation. When the pure lines were crossed, they produced an F1 generation. Once Mendel was sure that his lines were pure, he began his experiments. He started with a single trait, maybe round or wrinkled seeds. He crossed a plant that had round seeds with a plant that had wrinkled seeds. He called the offspring F1 for first generation. Use the following exercise to analyze Mendel's experiment to find out what happened when he crossed the two plants, or the two pure lines. So what he's doing is he's crossing two pure lines. So I'll draw that over here. So we had wrinkled and smooth, right? So we could say maybe wrinkled would be big W, and little w would be smooth. This is wrinkled. So what he did was he bred these two together. He bred these two together to see what their offspring would look like. So we call these the F1 generation. So let's do this little quiz. According to the theory of blended traits, what would be the result? So a blended trait, that would be the blended theory would maybe predict that the offspring would be a blend of wrinkled and smooth. My computer doesn't want to. There we go. Looks like my wife's trying to get a hold of me. Teaching. Can't talk. Boom. Mrs. Kirby. All right. All right. First generation, when the two plants, put on airplane mode. There we go. No more interruptions. When two new plants grew, every one of them had a smooth coat. The wrinkled trait had disappeared. What conclusion could be drawn? All right. I would say that we need more information. We just know that the smooth ones showed up, right? The smooth showed up when you mix the two, but we didn't get any wrinkled. I think we need some more information. Let's see. All right. When our next cross, let's fill this out. First cross, the phenotypes, right? They were all smooth, right? And the genotype, well, let's think about this. The genotype, so the wrinkled, we might want to switch these around actually. Let's say that the big W is smooth. And the WW is wrinkled. Okay. Oh, actually, let's go over here. This is actually, I forgot about this. This is actually pointing over to these pictures, tall and short. So let's look at these actually. So we had a cross between a tall and a short, big T, little t. And when they were crossed, the phenotype were all tall. There we go, tall. And the genotype, okay, well, let's think about this. If the tall plant only had big T's and the short plant only had little t's to give, that means that all of these plants have one big T and one little t. One big T, one little t. One big, one little. One big, one little. Which means the genotype of the offspring are all big T, little t. There we go. All right. So now we're on page 7. When the two F1 plants were crossed, the disappearing trait reappeared. So this was our parent. This is our first generation starting, right? This was our F1 generation, right? And this would be our next generation. After disproving the theory of blended traits, Mendel did another set of experiments. He allowed the F1 offspring to self-pollinate. When the offspring of the self-pollinating F1 generation grew, the results surprised Mendel. They did not fit any theories at the time. We're going to use this exercise to figure out what happened. All right, so now we're looking at the F2 generation. All right, the parents of the F2 generation were the F1 generation. This is called the F2. All right. So let's look back on the little thing on Apex. It says when the new plants grew, he noticed an interesting thing. The wrinkle trait that had disappeared in the F1 had reappeared in the F2. What conclusion might be drawn from this? I would say that wrinkle trait was there in the F1, we just couldn't see it. The F2 generation, there were 5,474 plants with round coats, 1,850 plants with wrinkled, for a ratio of about 3 to 1. What could be concluded from the number of plants used? All right, so from just concluding from the number of plants, that's the question that's being asked. I'd say that his data is probably reliable. He did this experiment a lot. All right, let's go to page 8. So Mendel hypothesized the existence of recessive traits. So remember recessive traits. We talked about those earlier in a previous study guide. When Mendel crossed the F1 generations, he saw the disappearing trait reappear. He concluded that there must be some sort of F1 generation determinant. He concluded that these heredity determinants exist in organisms from generation to generation, even if the trait isn't seen. He called these traits recessive, meaning that the trait seemed to recede. So we'll use this little review down here, which best describes the question Mendel was trying to answer. So looking at this a little deeply, I'd say that he's looking at the pattern of how the traits are passed from parents to offspring. How did he collect his data? All right, so he crossed the pea plants and observed the traits of their offspring. Why was he so successful? He used the scientific method. What was the purpose of the pure lines, right? Pure lines were the control group. Why were his experiments valid and reliable? It's because he repeated the experiments many, many, many times. And did his results support either of the hypotheses? So no, they did not support either hypothesis at the time, the blended trait hypothesis or the acquired characteristics. All right, let's go to page 9. All right, and before I start doing that, let's fill out the phenotype of the offspring on here. So we're going to do this one right here, phenotype of offspring. Phenotype of offspring, well, the phenotype is what you see. So we have tall, 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 and short. So we have tall and short, and actually a 3 to 1 ratio. Now let's see. Go a little further. Okay, just looking at the apex. The genotypes of the offspring, well, we know we have tall and short, all right? So since short's there, we know we have little t. But also, if you remember, if we have big T's and little t's, in the previous generation, the F1 generation is going to give the F2 generation big T's and little t's. So what you will find when you cross, we're going to do these a little later. We call these Punnett squares. You got big T's and little t's, big T's and little t's. You get offspring that are a combination of homozygous dominant, homozygous recessive, and heterozygous. So the genotypes are going to be big T, big T, big T, and little t, little t. You get a little bit of each genotype in there. Okay, so let's go back to page 9. Page 9, Mendel's laws of heredity. Mendel proposed that each trait has several forms that could be passed down. After many years of many experiments, Mendel analyzed his data and proposed several rules to explain the passing of traits from parents to offspring. His results were reliable because he had done the experiment lots and lots of times. In some ways, the choice Mendel made about which traits to study was a lucky one. All of the traits that he chose had a clear, dominant, and recessive form. This is not always the case in genetics. In addition, the traits were all located on different chromosomes, which is something he could not have predicted. This is significant because genes on the same chromosome do not always follow the patterns he discovered. All right, so let's go to page 10. So principles of segregation, or the law of segregation. So Mendel's work went mostly undiscovered until after his death. It wasn't until the early 1900s that it was rediscovered. Several scientists began to combine the results of his work with research on meiosis and chromosomes. The patterns and data Mendel recorded are easier to understand if seen through the lens of meiosis. This is because meiosis causes the patterns of inheritance that Mendel observed. Mendel's principle of segregation is based on alleles and chromosomes. Although he didn't know about chromosomes, he said that peas have two versions of each trait, and only one version is passed on in each egg or sperm. How does this affect genotype and phenotype of the offspring? So let's take a look at this video.
During meiosis 1 and 2, parent cells divide and form 4 daughter cells, or gametes.
What do you know about the gametes of the YY parent? All right, so the YY parent can only give capital Y parent. It can all of its sperm cells only have the allele capital Y.
All of the sperm contain the big Y allele, and all of the eggs contain the small Y allele. When these plants self-pollinate, the resulting offspring will be heterozygous for the trait being studied.
So if yellow seeds are dominant over green seeds, what will the color of the heterozygous plants be? All right, well, if yellow is dominant and it's heterozygous, all right, all yellow. Ratio of phenotypes, so let's go to page number 11. All right, but before that, we'll fill out the law of segregation part. So how many alleles for a trait can be passed on from one parent? One allele from each parent gets passed on. So how many alleles does an offspring inherit? One allele from each parent. All right, ratio of phenotypes. So the 3-to-1 ratio observed by Mendel can be explained using chromosomes. Each plant in the pure lines of Mendel's peas carry 2 copies of the same version of a trait. Thus, every member of the pea or parent generation is homozygous. For every trait Mendel studied, the F1 generation showed only the dominant trait. This is because every member of the F1 generation is heterozygous. Each one has a dominant and recessive allele. That's what we looked at up here, right? The parent generation is homozygous, right? It's homozygous. Big T, little t, little t. And when those come together, this parent can only give big T's and this parent can only give little t's. So that means that the F1 generation is all going to be heterozygous. But in the F2 generation, Mendel noticed a pattern in the number of offspring of each phenotype. The ratio was always 3 dominant to 1 recessive, or 3 to 1. For example, when Mendel was looking at tall and short plants, there was always, and we saw this right here, 3 tall and 1 short. To understand how this works and why it produces a 3 to 1 ratio, it helps to look back at the process of meiosis and follow the chromosomes from the parent cell to the daughter cell. So let's look at this.
What happens when these cells go through meiosis? Unlike pure lines, heterozygous plants will produce 2 different gametes. The 2 alleles, big Y and small y, separate during meiosis to make daughter cells with either a big Y or small y allele. Depending on which sperm fertilizes which egg, the genotype of this offspring could have several different alleles. Combine each egg with a sperm to observe the different possibilities for the offspring.
All right. So you can drag them over from left to right to combine each of them.
These are the different offspring that can be produced. According to the rules of probability, there is an equal chance of any of these events happening.
So how many different alleles are present in the egg and sperm cells? So there's the alleles. There's 2 alleles, big Y and little y. How many different genotypes? There's 3. There's homozygous dominant, homozygous recessive, and heterozygous. And that leads to 2 possible phenotypes, green and yellow. What is the ratio? 3 yellow to 1 green. Mendel's results led him to ask more questions. Mendel discovered a pattern to the inheritance of a trait. Through his single trait or monohybrid crosses, he determined that he could predict the outcome of crosses as long as he knew which trait was dominant. But what if Mendel crossed a pea plant that had yellow, round seeds with a pea plant that had green, wrinkled seeds? In other words, would the pattern of inheritance for one trait depend on the pattern of inheritance for the other? So what he was asking is, would one trait affect another trait for inheritance? So let's go to page number 13. So different traits are actually passed independently of one another. To find an answer to this question, Mendel conducted experiments called dihybrid crosses, in which he followed 2 traits. Mendel crossed a plant with round yellow seeds and one that had wrinkled green seeds. All of the offspring had the dominant traits, round yellow seeds. Mendel then crossed these plants and found that a plant that had yellow seeds was not more likely to have round seeds. In other words, the plant was just as likely to have yellow wrinkled seeds as it was to have yellow round seeds. Color and seed shape were passed independently, and this is known as the law of independent assortment. So we'll write that down here. So each trait is passed on separately from each other. What evidence support this law? And that's that different traits do not affect each other when going from generation to generation. Go to page 14. So, Mendel's laws of heredity were results of years of great ideas, careful record keeping, and understanding the importance of data. However, finding one of those laws, the laws of independent assortment, was also a great deal of luck. The garden peas that Mendel chose were a perfect choice because they reproduce and grow quickly, they have easy to distinguish traits, and have produced large numbers of offspring. But Mendel didn't know was that alleles for 7 traits that he chose to study have to be located on different chromosomes. In addition, each trait had a clear dominant recessive form. Traits that follow these patterns are called Mendelian traits. So, Mendelian traits, traits that are controlled by one gene. They have a dominant and recessive form. All right, so let's go to page 15. Traits that do not follow Mendel's patterns are called non-Mendelian traits. Mendel's theories don't work for every trait. There are other traits called non-Mendelian traits that do not follow his patterns of heredity. Traits controlled by more than one gene, alleles that are not clearly dominant, and genes that are where there are 2 alleles, all lead, or more than 2 alleles, all lead to non-Mendelian patterns. Look at this graph of women's shoe sizes. So we look at the graph, and the shape of this graph is called a normal distribution. A normal distribution basically means that there's a lot of data in the center of the chart, and then it tapers off on both sides, right? So you would see this with height of people. There's a lot of people who are of average height, and then there's not very many people that are 4 feet tall, and there's not a lot many people that are 7 feet tall, right? So there's less people on both sides, and the same thing with shoe size. For women's shoes, I guess there's lots of people that wear a size 5, and not so many that wear a size 1, not very many that wears a size 9, based on this chart. So the question is, if foot size were a Mendelian trait, what would you see when you compare to the if you compared it to the shoe size chart shown here? All right. So if it was a Mendelian, there would be a clear dominant and a clear recessive, which means there would just be a big size and a little size. Based on the graph, how would you describe the distribution of shoe sizes? Well, I would say some sizes are very small, some sizes are very large, but most are in the middle. How could this normal distribution of sizes be explained based on the idea of genes? So, shoe size must be controlled by more than just one gene. What is another trait that is controlled by more than one gene? I would say skin color. All right, so skin color comes in a vast majority. Some people have very dark skin, some people have very light skin, and a lot of people have skin right in the middle. So skin color is a trait that is controlled by more than one gene. So let's go to page 16, and then let's fill in, describe non-Mendelian traits. So traits that don't fit or don't follow Mendel's patterns. Usually they have maybe more than one gene that controls them, or more than 2 forms, 2 alleles, or no clear dominant or recessive. All right, so page 16. Mendel is known as the father of genetics. He discovered how traits are passed from parents to offspring. He developed theories about alleles and genes, even though he didn't even know that genes existed. Because of this work, Mendel is known as that father of genetics. Once scientists discovered chromosomes and DNA, they were able to connect this new information to Mendel's work. Since then, the field of genetics has taken off, and here are some current applications of Mendel's work.
Some scientists study genetic abnormalities and how these abnormalities cause defects in the structure and function of cells. Scientists use genetic information to understand how species are related and how they developed over time. Scientists are manipulating genes to produce plants with specific traits, like drought resistance. Many feel this is important work to feed the growing human population. In 2003, the complete DNA sequence contained within the 23 pairs of human chromosomes was published. When the sequence was printed onto paper, it took up an entire bookshelf.
All right, let's go to our last page. So that is it. That is our study guide for 7.3.1. I hope that it wasn't too long for you. It takes a while to go through these and fill them out, especially trying to explain these as I go along. You should be able to pause it. All right. I'll do this at the end. I'll let you pause it there to get some of that information. Pause it there to get some information. Pause it there to get some information. And you could pause it there to get that info. All right. So this is Mr. Kirby signing off, and I will see you later. And we will be doing, let's see what the next one will be. The next one will be, we're at 7.3.1. The next will be 7.3.3.